go. Okay. Go back to this. Right.
Looks like we have a quorum.
Yeah, I think we've got our whole panel, which is fantastic. And let me stop sharing for a moment there. Um, and it is now 8 o'clock. So, Alan, you want to start us off?
Of course. Thank you everyone for making the time to our first of three clinician scientist webinars hosted by the ARO as well as the Triological Society. Aaron Schneider has put together a wonderful panel. So let him introduce the group. Thank you all for making it.
Thank you, Alan, and thank you for all of your legwork and getting this ARO committee off the ground. I have to say that I think much of the committee's work has been underway over the course of the last year, and I'm really honored to be a part of this panel and to be joined by a number of distinguished panelists who represent really the many faces of clinician scientists working across otolaryngology. Um, I also want to just briefly say thank you to Yuri Agrawal, who was our ARO president last year and hosted a presidential symposium focused on clinician scientists, and I think really sparked a lot of the conversations that remain ongoing. So in terms of format, I'm going to introduce our panelists. I have a number of questions that we'll go through and have hopefully a lively discussion over the course of the next 45 to 50 minutes. If there are questions from the audience, I ask that you place them into the chat. And we hope to be able to get to those questions towards the end of our session. And in respect for everybody's time, we'll make sure that we end right around the 1 hour mark, which for those of us on the East Coast is right around 9 p.m. So I will begin by introducing Aaron Ford, who's at the top of our speaker panel on the right side of your screen. Dr. Aaron Tord is a neurotologist scientist and an associate professor of otolaryngology at the University of California at San Francisco. His clinical interests include cochlear implants, tumors of the skull base, and surgery for chronic otitis media. And his NIH-funded lab investigates tumor biology, the genetics of disorders of the head and neck, and the development and physiology of the organs of hearing, including the tympanic membrane. In 2022, he went on partial leave from academic practice to serve as the chief scientific officer of Akuos, which is now a wholly owned subsidiary of Eli Lilly. And his focus at Akuos is on developing gene therapies for hearing loss. So welcome, Aaron. Thank you for joining us. Eric Holbrook is a rhinologist scientist and associate professor of otolaryngology at Harvard Medical School. He serves as the division director of rhinology at Mass Eye and Ear, where his clinical interests include diseases of the nose and sinuses, endoscopic approaches to the skull base, and smell and taste disorders. His collaborative research with James Schwob, a PhD basic scientist at Tufts, focuses on the mechanisms of olfactory function and methods for the reestablishment of olfaction following loss of smell. And he provides a critical clinical context for collaborative translational work in olfaction. So welcome, Eric.
Thanks, Aaron.
Susan Emmett is an otolaryngologist scientist who serves as an associate professor of otolaryngology at the University of Arkansas for Medical Sciences. She treats patients with otologic disease and has a focus on hearing loss prevention. She is the founder and the director of the UAMS Center for Hearing Health Equity. And Dr. Emmett studies novel pathways for hearing loss prevention and applies digital innovations such as mobile screening and telemedicine to extend access to hearing healthcare to rural and underserved communities. Her work is both local in Arkansas, at a distance nationally in Alaska, and also across the world. She routinely consults with public policymakers in Washington, D.C. to guide improved access to healthcare for underserved populations. So, welcome, Susan. And last but certainly not least is Dr. Rona Herzano. Dr. Herzano is a surgeon scientist who specializes in the diagnosis, treatment, and restoration of hearing loss. Her research focuses on preventing hearing loss and developing innovative strategies for hearing restoration using multi-omic approaches to uncover key regulators in inner ear development. Her work has identified and characterized several key transcription factors essential for hair cell development and advanced our understanding of age and noise-induced hearing loss and identified drugs for repurposing to prevent acquired hearing loss and also identified and characterized sex-based differences. Dr. Herzano has founded the Gear Portal for sharing omics data in hearing research and HearShare Consortium to advance auditory and vestibular clinical data sharing. She is committed to mentorship and actively fosters the next generation of researchers and clinician scientists. She also holds leadership positions in several prestigious journals and is the president-elect of this organization, of the ARO, next year. In February of 2023, Dr. Herzano transitioned to the NIH, where she is a senior investigator and chief of the newly established neurotology branch at the NIDCD. And while in this leadership position, Dr. Herzano continues to maintain a part-time clinical practice at the University of Maryland School of Medicine as a clinical professor. So I hope you're able to hear that we have a number of fantastic and very successful surgeon scientists who come from a number of different backgrounds today. And I hope that we will all learn about the many ways in which we can pursue both research and clinical interests simultaneously. With that,
the webinar begins.
Yeah, I got that on my computer, and I got. And if I could just ask for everybody to mute as they join. Thank you. I will show one quick disclosure slide. There are several disclosures, but no conflicts related to this discussion. And with that, we'll start right into our conversation. I want to begin perhaps with Dr. Emmett. If you could tell us a little bit about your career path, what went well for you, and perhaps what maybe you would have done differently.
Sure. Thanks, Aaron. So as Aaron mentioned, I do disparities research on hearing loss, and I work in both domestic populations and internationally. And my path to where I am now, where I funded a large NIH-funded research center, is I worked in policy before I ever went to medical school, both at the NIH and Capitol Hill. And so that laid the groundwork of the lens that I approach all of my research through, because our ultimate goal is to change policy and ultimately change lives. And we don't stop at the paper stage or the ending of a grant. And so I approached my medical education in somewhat of a different way, because I knew that I wanted to go full circle back into policy from a research angle from the very beginning. And so I ended up spending a year of medical school doing research in East Africa to make sure that boots on the ground experience, this is really what I wanted to do. And it had nothing to do with otolaryngology, because there were no opportunities at that time. Really, no one was doing clinical research within our field internationally that I could tap into. So it was in a different field altogether, but laid the groundwork for confirming that's definitely what I wanted to do. And so I chose to train at Hopkins, where I could both do my otolaryngology residency, but also spend a couple of years in the School of Public Health, getting an MPH, doing a postdoc, and working with nutrition interventionalists that did large pragmatic randomized trials, primarily in South Asia. And so that's where I got my experience with how to run randomized trials. That has really informed where I am today, because now we run, our center has a couple of trials that inclusively have about 20,000 children in them and 100 sites in 2 states, 3 regions of rural Alaska that span essentially the equivalent of the entire eastern seaboard in terms of geographic distance, as well as about to 15,000 children in Appalachia in a trial. And we have some studies going in Arkansas as well. And then our international sites right now are in Sierra Leone and South Africa. So all of that background in terms of policy work and a really solid grounding in public health research methods is what was needed to set me up for success to be able to do this type of work.
Thank you, Susan. Anything offhand that you maybe would have thought about doing differently, knowing where you've come and the course that you've taken today?
I think so, because the experiences that I had early on were really fundamental to shaping how I approach the scientific questions that we study today. So while it may sound like a meandering path, it all sort of fits together, and there's a purpose behind all of it. So it took a while to go through all of that process, but it was definitely worthwhile and gave me all the building blocks that I needed.
Great. How about Eric, Dr. Holbrook? How would you describe your career path, and how did you get to the point to where you are now?
I don't know. I feel like I had a kind of a unique path. It was maybe not so unique. I mean, I feel like I started out in medical school being introduced to this science of olfaction and was just blown away as like a neuroscientist. That was my interest in undergrad, that these nerve cells could regenerate. That's what really got my interest in the whole clinical aspect of smell loss then came after, really. But I was able to form this relationship and get a master's degree and do this extra time research during medical school. And the mentor that I had then, became my mentor as I got a position at Mass Eye because he took a position at Tufts while I was in residency. But in my residency, I spent time with another olfactory research person that also became my mentor, and did my fellowship under a person, Don Leopold, who was, very well known for clinical aspects of olfaction. So all that part of my training was really great because it kind of rounded me out. Having a position at Mass Eye and then being able to then do the basic science type research I was interested in. Jim Schwab's lab at Tufts was huge. I mean, it was very fortuitous that I was able to get this position and also have somebody nearby that was doing research that I knew and was in the area. I was interested in continuing the research. So that was great. I guess thinking about like what kind of pathway would I have taken that might have made a difference? I always wonder, Jim Schwab, when I was a medical student, asked me if I wanted to go on past the master's that I was doing for taking a year off and doing research and doing a PhD. I don't know. I don't know what the answer is to that. Would it have given me a little bit more experience in doing something like writing grants or, like putting together projects? I don't know. I mean, I feel like the time that I spent might have been longer in terms of kind of having the opportunity to do that during my career. But I always kind of wonder if getting extra education as a PhD would have made a difference. But there's a lot of things during my career that, you always kind of look back and you wonder, should I have spent this time doing this? It's more time management, I think, how you manage your time outside of research, your clinical time, your administrative time, your time with societies and things like that. If you add it all together, there is some distractions that at least I question whether or not they were good choices.
Well, I think I hear something both from Susan, from you, and from Eric that I think probably speaks to a lot of us, and that's early exposure, probably during the medical school phase, can have a lasting impact. Um, but the pursuit of a PhD may not be for everyone. Eric and myself and Susan are surgeon scientists, but do not have PhDs, have other advanced degrees. But Aaron, you did a PhD during your medical school, and obviously that was, I think, heavily influential for you in terms of choosing a path as a surgeon scientist. Can you tell us a little bit about how you got to where you are now?
Sure. As mentioned, when I was coming out of college, I knew I loved science and I knew I loved medicine, and I had indecision, so decided to enroll in an MD PhD program. And I had worked primarily on in my PhD on tumor biology and liver development and liver tumors, but hadn't really been exposed to the world of neuroscience until I took a neuroanatomy class in medical school. That was fascinating to me. And then later, when I did my clinical rotations, I was exposed to cochlear implants and thought, wow, this is like the coolest thing I've ever seen. I should probably go do that for a living. And so thus I decided to do that. And when I went off to residency, I still had it in my mind that I wanted to be a scientist with perhaps the majority of my time. And I selected a residency in large part based upon going to a place that I thought would be clinically very heavy and give me a good surgical training, because I figured if I was doing surgery with the minority of my time, I didn't want to do remedial surgical work or do harm to my patients by being not quite as good as somebody more experienced. But I also wanted to be in a place that had, a very strong scientific community with diverse experiences, which indeed Mass Eye and Ear was, which is where I went to residency. While I was there, um, there were some very exciting things going on outside of the department with regards to sequencing and high throughput sequencing in particular. And there was a place called the Broad Institute just across the river that was one of the first places in the world to really get access to the sequencing technology. And having a background as a tumor biologist, there were a variety of questions I was very, very interested in. And so I approached PIs there and worked with a mentor at the Broad Institute outside of the field of otolaryngology, and did a lot of work on sequencing at that time and picked up some computational skills that I think were helpful. And he encouraged me to work on head and neck squamous cell carcinoma, which I did. And then when I completed my residency, I did a fellowship in neurotology because I had always been fascinated by the ear and with cochlear implants in particular. And then I went off and started my academic career at UCSF, where I was starting up a lab, spending in principle about 40% of my time doing clinical work and 60% of my time doing research. One thing that did happen, and this is a cautionary tale for surgeon scientists everywhere, after I came, one of my only partners left, and I was left as the only full-time otologist at UCSF 4 months into starting my job. And my department chair came to me and said, "Hey, your lab space is getting renovated anyway, and we're in kind of a crunch here. Do you think you could go up to full-time clinical? And I promise I'll make it up to you later." And I said to him, "Okay, I'm happy to do that, but with two conditions. One, when the time comes that promotions are going to be happening and things like that, that you won't harass me that I didn't have enough papers out or grants out, or at least that I was only 1 year behind or 2 years behind." And the other thing I said is, let's pick a defined end date when this is going to end. And I'm happy to do this to help out. I know you're in a crunch right now, but we're going to pick a defined end date and get back to our original deal, which indeed he was true to and very supportive of. So I launched my lab to work mostly on cancer, then started getting into the ear. I'm very much an opportunist with regards to the questions that I'm asking and answering. I'll work on anything that I think I'm uniquely poised to be able to make a contribution, and I don't feel particularly limited on what that is. And so about 8 years in, or about 6 years in, I started getting involved with consulting for some companies. They had reached out to me to ask for some help, and I was invited to serve on the scientific advisory board of this company, Akuos, that was making gene therapies for hearing loss. And then a couple of years ago, they came to me and said, "Hey, can you spend a little bit more time here? We're entering a critical phase with our gene therapy trials and new drugs." And I said, "Okay." And so off I went. And now I've been basically spending most of my time now out in the world of biotech at that company, and with still the minority of my time here at UCSF, but shifting back and forth a little bit.
Thank you. I think you touched on quite a few points that I think are really salient. One of which is that in the clinical context, there's never a shortage of patients. And the clinics will always be full. And so I think your point about having a conversation in terms of defining your ability to really reestablish a schedule that works for you as a surgeon scientist is really critical early on. And also, I think very honorable that your chair was willing to abide by that agreement, because sometimes it's very easy to ramp up the amount of clinical work that you do, and getting back that research time can sometimes be a challenge. For those of us like myself who went down the K08 pathway, the federal government helps us a bit to define exactly how much time that we need to have in the lab. And so we do have a paper that we can hold up and say, really, I need to be 50%. So that can be a helpful mechanism by which we can define that time split. But Rana, I'm interested to hear a little bit about your career path, having spent time and risen to the rank of full professor at the University of Maryland. You made a recent switch to be an intramural investigator and direct a new neurotology branch. How did that come about? And tell us about the really great decisions that you were able to make along the way.
Thank you for having me. So I never wanted to be a researcher. I was a national team athlete, then I went to medical school to fix knees. And I didn't know anything about research. I knew everything about track and field. And it was a 7-year program, and in the first summer I came back from a long trip, and it was a message on my receiver from a friend who said that he's in this amazing cancer biology lab that he thinks I'm going to really love, but he is really bored at. So he told him that he's transferring his scholarship to me because he knows I'm coming back the next day. And I had a scholarship for 4 weeks to go there. And I was really excited because, I've never been in a hospital really except for visiting friends. So it was an opportunity to do research in a cancer biology lab. And by the end of the 4 weeks, I gave a draft of a review to the PI. I learned so much. And PI met with me and asked if I wanted to transition to the MD PhD track. And very naively just asked, what's a PhD? And when I heard what it was, I thought it was the most awful thing I've ever heard about. It just didn't make sense. You stop, you need to make a discovery. You can't go back until the discovery is cemented and published first. And then all your friends finish medical school. So I went back to medical school, and the next 4 years, I kept going back to that same lab, other lab, not that one. I looked for a different lab, and I chose a lab that was led by a scientist, an Israeli American scientist that was interested very much in hearing. And she's now she's a very famous researcher. That's Karen Abraham. She had a lab, and she still has it until Aviv University. And she agreed to take me every summer without making me do a PhD. And it was about 4 years into doing that, and actually after already cloning a mouse model as a medical student, that I had this enlightenment where I realized that I could stop medical school for a few years and actually do even more. And the human genome sequence and the mouse genome sequence were coming to completion, and microarrays were coming out, and they had this mouse model for a gene that when it's mutated in human have hearing loss, and in the mice, the hair cells are formed, but they can't survive even 3 or 4 days. they immediately die, which means that anything this gene turns on or turns off is just critical for hearing. And I just couldn't bear the thought that someone else is going to find out what it turns on or off. So I switched to the MD PhD path, and a few months later, Karen suggested that I switch to the shared program with the NIH and the women health track, which is kind of funny because we close circle like many years later. At the time, it was just because women had ears too. And I spent 3 years traveling between Tel Aviv and Bethesda. I was mentored in Bethesda by Tom Friedman and Matt Kelly. And I met there Andy Griffith and Tomoko Makishima and some other surgeon scientists that you might know. I learned about residencies in the States. I learned that it's possible to be a surgeon scientist. I met here at the NIH neurosurgeons that were surgeon scientists because, at least in my medical school, everyone told me that if I wanted to be a scientist, I really shouldn't be a surgeon. And when I went back to medical school, I knew I wanted to be an neurosurgeon. And if you need to be a cardiologist to be a neurosurgeon, I'll be a cardiologist, but happen to be together with the nose and the throat, so that was fine too. I chose my residency based on, I interviewed in 8 places, and a lot of places I was asked if I'm more a surgeon or more scientist, but when I came to University of Maryland, which was ranked awful at the time, it was just had multiple changes of department chairs. There was this new chair there that was like a surgeon scientist, and it was super welcoming. And he basically said, you're a surgeon scientist, I'm a surgeon scientist. I know what it takes to make a surgeon scientist. I'll support you. And when you finish your residency, I'll hire you as faculty. And, I walked around, I walked around there and saw people were very, very nice and welcoming and decided not to take the chance on them. And the department really got a lot better over that time. I went from being not ranked to being ranked in the top 10 in the country. And he was true to his word. He gave me, this first 6 months, I decided not to do anything. And then he offered me a technician and some help. And I was able, in some protected time, and I was able to do research throughout my residency. And he taught me how to write grants, and I got funded every single year of my residency. And so by the time I finished my residency and was interviewing for fellowships, he made a competing offer of a fellowship at the University of Maryland. And I think that when you think about things that, what would I've done differently, right? That's one point that you never know which road would have been better to take, right? I stayed at Maryland. I was mentored by an absolutely wonderful clinician. I got a very good clinical training. But I saw only one way to do things. Having said that, I was able to build my program, and within not even 3 years of finishing my residency, I was both ARO1 and DoD funded. So there are pluses and minuses for everything, and I think it took me about 4 or 5 years after residency to feel that I'm as good a neurologist as anyone else, or definitely a hearing restoration surgeon, even though I did all of my training in one place, and I didn't go to another place and train with another full set of clinicians. And so that's one of the things that you never know what was the right thing to do. At Maryland, it was a great environment. I was there from 2011 when I finished residency until 11 years later, when I left as a tenured professor. And I never interviewed anywhere else. I never wanted to go anywhere else, and I'm still there because I still like University of Maryland. My department chair really wanted me to be successful as a clinician scientist. And the first thing I did coming out of the fellowship was he asked me, what do I want my time split? And he offered 60% clinical, 40% research. And we negotiated what Aaron just mentioned, 40% clinical, 60% research. I took a pay cut for the first year to be able to get that split. But I think that having that split was critically important. And then, you know, it's when you're starting off, it's very easy to basically want to do more. And he'd call and be asking, what I'm doing. And if he hear that I'm in the office signing dictations and whatnot, I'd be told off and be, sent back to the lab. I could never be caught on a lab day in the clinic for any possible reason. I'd be sent immediately back to the lab and explain that, there aren't that many surgeon scientists, and I will not be successful if in my research time I'm spending it in a clinic. And for that and for 1,000 other reasons, I'll forever be grateful to Scott Strom, who was my mentor and still is, because he really knew how to keep a surgeon scientist that is very tempted to generate more RVUs and be a team player on track. The decision to go to the NIH was not trivial because NIH was not known for a lot of clinical work. And as a clinician scientist, we all want to be clinically heavy as well. But NIDCD got new leadership a few years ago, and the new leadership became very focused on translation. So Dr. Tucci from Duke became the new director, and she started changing the grant application mechanisms, the focus of the NIDCD to be a lot more focused on translation, and then decided she wants to have a neurotology branch that will be, if you want, a greenhouse for translation, and at the NIH. And we spoke several times, and I learned about this position, and I got convinced that the intents are genuine. And I met everyone that was there at this time, and I decided to interview for the position. And I was extremely fortunate to be able to be ultimately elected for this position that really allows me to do research at a very high level, collaborate, but also maintain a clinical presence at University of Maryland.
Thank you, Rona. I think you mentioned a couple of really important points, including the role of mentorship and early, administrative or chair level support for surgeon scientist split can really be the difference between a successful and a very challenging surgeon scientist career. I want to shift gears a little bit and ask about specific recommendations for some of our early career clinician scientists and trainees. I think there have been, sounds like the pathway for many of you guys so far seemed to be very streamlined. It was one step leading to the next and getting you ultimately to success. But, Eric, if I could ask, do you have any specific recommendations? What would you say, briefly to a early career, say a resident who really is interested in becoming a clinician scientist?
Yeah, I mean, Rona hit on some important things that I was thinking of too. I mean, she sounds like she had a very, very, very good mentor that understood the whole clinician scientist kind of role, whereas the person I did my research with and really looked to as a mentor for research was not a clinician scientist, but a PhD, MD PhD, but did all his work in the lab. So not knowing, I don't think he really had the idea of what it takes to stay away from building your clinical practice to the point where it's taken over a lot of your time, right? So I think time management is huge. You really have to carve that out in the beginning and hold true to the time that you're going to spend on research versus the time you're going to be in the clinic or administrative or societies and things like that. And I think that's where you're going to have your most success. But we're also kind of neglecting what the definition of success is. True, like how do you want your clinician scientist kind of hat to be? Like, how into the science do you want to be? Do you want to be a collaborator? Do you want to really drive the research? Those are all questions you have to come into early on and really kind of get the idea of what your role is going to be in research. And that's a hard question sometimes to answer without a little bit of experience first. So the whole time management is a big deal, I think, if you really want to do basic science research. Um, because once that goes away, I don't know how Aaron did it, but once once it's taken away from you, that kind of protected time for research, it's hard to get it back again, you know.
I think that definitely. Susan, what would you say to a junior faculty member or even a resident who's considering a career as a clinician scientist in terms of really what recommendations for launching a successful career?
There's a couple of things that have already come up that I'll highlight, and then a few more. I think one is making sure that you choose wisely where you train and where your first faculty position is, because it's really critical to have someone who wants to set you up for success and understands what it means to build a successful surgeon scientist career, because that makes all the difference in the world. And they don't have to be in your field necessarily, but they need to be fully supportive of what you want to do. And I had the benefit of that in the late John Naparko when I trained at Hopkins. Really believed in what I was trying to do, which was extremely unconventional at that time. And that would have been very hard to accomplish in other places where I didn't have that kind of support. And I think there's also looking for mentorship broadly. Your mentorship doesn't have to be in otolaryngology. Sometimes the best mentors, depends on what you want to do, but sometimes the best mentors are actually in other fields. And they may or may not be clinicians. I mean, a longtime mentor of mine was actually an ophthalmologist who ended up being the dean of the School of Public Health at Hopkins for 15 years. And I learned from him both sort of how to go about this process and also learned a lot of science from him. So thinking sort of broadly about mentorship. And then the third thing I would say is be really open to opportunities. So I knew what I wanted to do early, and I was pretty dead set that all of my research was going to be international when I was in training. And that's the pathway that I was headed down until two mentors within 72 hours of each other both told me I needed to meet a certain surgeon in Alaska because of interesting, innovative work he had done in telehealth to address the major issues with chronic ear disease that happen in circumpolar populations around the world. Well, this wasn't on my radar screen at all. I'd never been to Alaska. One thing led to another. I listened. I met this person. Ended up spending the last couple of months of my residency working with him. Ended up joining the Indian Health Service and spending time there. So I understood the clinical system, built research relationships, and actually got a $2 million PCORI award funded before I ever finished residency because of all of that, because I listened to these two people who suggested that I talk to someone in a state I'd never been to. And that has dramatically influenced my career to this day because I do a lot of domestic disparities work, not just international. I do both. So I think be open because sometimes unusual suggestions or seemingly unusual suggestions might have a big influence.
Absolutely. Aaron, you've run a lab and have had a number of motivated students and even residents come through. What are you talking to them about? Or, what advice do you give them in terms of a career as a surgeon scientist?
Yeah, I think the first piece of advice, which I think is the most important, is figure out what you really care about and what you want to do and what you're excited about doing. Because in particular, as an otolaryngologist, we don't have the same kind of traditions as surgeon scientists as like being in hematology oncology, for example, where you can go and be one of 20 fellows at a big center that have established pathways and where lots and lots of people have done it. So what we do obligately has to be a little bit more bespoke, and you need to sort of create and cobble together what you think the opportunities are and have a vision of a pathway that you want to go and go in that pathway. And you're not going to be willing to go in that direction unless you have a specific vision that you really care about and want to articulate. The other aspect of that, though, is that is that it's unlikely, if you listen to what any of us have done, none of us did like a single program that went linearly through a pathway that if we just kept on going and did that program, you sort of get to the end. It was all, very different things and chance encounters. So as a consequence, I think one thing that's important is that as you're, say, for example, looking for faculty positions, you need to articulate a vision and how you're going to go about doing it, including the scientific vision, but also including, how are you going to go get funding for this, and where is the money going to be coming from, and how exactly is it going to work out, and articulate that vision to wherever it is that you want to go next, in order to say, this is how I think it can work. This is how I think the plan can get executed. And if you've got a realistic vision that also includes the pragmatics and things like money, for example, you're much more likely to be able to get that, so that you're presenting an idea to somebody. And touching off what I think both Susan and Rona have said as well, if you have a specific vision and a specific way that you want to go about doing things, you're much more likely to find somebody who's willing to give you either sort of a thumbs up or thumbs down on that. You want to find somebody who's going to be on board with that particular vision and share enough of that vision that they will be supportive of going through the sort of ups and downs of it and doing what it is that you want to do. I think it's much more difficult if you sort of go out looking for jobs and they might be looking for a very busy clinical otolaryngologist and trying to cobble together something that, when what they're looking for is not exactly what your vision is, I think is a much more uphill battle.
Rona, having been at Maryland, you mentioned that you had great support from a clinician scientist mentor early on, but that perhaps it wasn't the same type of environment as a really large academic center that has a lot of basic scientists that are around. How were you able to chart a course early on, and what do you advise to your trainees, students that come through the lab and that you work with from a clinical standpoint now?
Sorry, every time we start talking, the light turns off. So these are excellent questions, and I agree with everything everyone said. In particular, Aaron, I think my first message to aspiring clinician scientists is that you have to love this. You absolutely have to love this. It's going to be years before you have help. And so you really have to want to do it. Like, I can't tell you how many years I was in the lab even as junior faculty at 6 o'clock in the morning already dissecting things, to be done with them by 8, to pass them on sometimes to a technician that totally destroyed what was done early on in the day, to then come back. But the other aspect of it is you can't burn yourself. So my rule was on rotations that like are head neck, I'd pretty much pack up the lab. I wouldn't even try to come to the lab. Maybe just, check on the mice twice a week, and not just to see that they're alive and, that the cages aren't overcrowded, because it's really, really important to pace yourself. This is sort of like a lifestyle. This is not a sprint. But you're right. When I was at Maryland, the only person that did ear research at the time was just moving to College Park. And I met with Scott after 6 months in internship. And he presented an absolutely amazing project he wanted me to do that ended up being a startup that ended up being extremely successful. And I didn't hear the word ear even once. So I basically said, I have not heard the word ear even once, and I want to continue being an ear researcher. And his response was, I'm not an ear researcher, but I can be your mentor. He's a head neck cancer biologist. And he gave me $5,000 and said, come back when you're done spending them and show me what you got. And when $5,000 is all you got, it's amazing what you can make happen with $5,000. And that allowed me to generate some preliminary results that were based on things that I had before. And one thing led to another. And when you show that you're really serious about things, people around you want to support you. And I had another person, Dr. Taylor, that's now our current chair, that ended up supporting a technician for me for several years and not asking even once to be on the paper. At one point, Scott took me to Claire Fraser, who was the head of the Genome Institute. And he basically said, I've got this resident in the department and I can't really scientifically mentor her, but it seems like she's doing things in omics. And that brought on a lot of opportunities. I met a lot of really interesting things, interesting people. And and when I had the idea for the gear, I met, I met a couple people there that thought it's not a good idea. And then I went other places and they didn't want to do it. And when I came back to the IGS the second time, they decided to try it out. And, the gear became a very central thing for the field. So there were a lot of things that, are almost random, but I think the one thing that is common to all of them was that I was just really passionate about the science. I was willing to put in the hours. I was really curious to figure things out. And in the path, I met a lot of really good people that were happy to support it.
Thank you. I know that both Aaron and Eric, you guys have both had collaborative labs, meaning that you've worked with other researchers, be it basic scientists or other labs across disciplines. I've done some of that myself, and I wonder, Eric, if you could just speak to what makes a magic collaboration, and sort of what are some of the challenges that are involved with a collaboration, where you're not doing everything yourself right within your own lab, but you're collaborating maybe across institutions even.
Yeah, I mean, the cross institutions, I was nervous about that in the beginning. Joe Nadol at the time was really good about fostering, this because of the limited space at Mass Pioneer, of course. So I think he was all for it, and I think that's held true. That most of us at Mass Pioneer that want to do research can still easily collaborate in another institution or without any I don't know, like side eyes or anything like that. It's actually probably encouraged to a large degree. Um, it does cause a little bit of paperwork struggle at times and figuring out like where the indirects and directs go and how they get kind of parceled out a little bit. But you can kind of ignore that just to get the science. Like if you're really interested in doing the work and the person that you're with is someone that you really believe is gonna help you do that work, it's pretty easy than otherwise. Um, it is always easier if your lab is right in the institution where you're doing your clinical work, so you can go back and forth pretty easily. So that makes it a little bit of struggle, but Boston's kind of a small place and Tufts isn't too far, so I made it work. It also separates you in some ways. It's kind of beneficial because you're not seen, right? So nobody can come and find you if you're in a totally different hospital and working in a lab somewhere where they can't find you. Um, so I mean, I found it to be not that much of an issue. And I think starting projects on your own might be a little bit harder because you do need that mentorship if you don't have the lab of your own to make sure that it is in line with what they're doing and the funds are available to do that unless you get them yourself.
Yeah. Well, and I think for many clinicians, it's perhaps a path of less resistance early on to join with a group that has, a lot of expertise in a specific domain and to provide that clinical context that they may be really thirsty or yearning for. Susan, I know you.
I mean, I think the bottom line is science is a team sport and it is better together. And yes, your grant administrators will groan when you've got 4, 5, 10 subcontracts like we often do. But you want that because you'll be able to do better science by working in large collaborative teams across institutions. And so don't let anybody push you away from that, because I think that's where the innovations really happen, is build those research networks, develop those relationships, and bring people together across institutions, even countries, and you can make, that's where the magic happens, I think.
Yeah. And the other thing I'll add to that, I couldn't agree more with Susan, but the other thing to add is that there are things that happen in other fields and in other groups that are very, very routine for them, that they've gotten very, very good at doing, that we have a lot less experience with, and which, when combined and sort of focused on the right particular question, could make a really big impact in a relatively short amount of time comparatively, particularly for things that just haven't really been applied and done in our field before. And really trying to take advantage of that expertise that other people have, you can find synergies very, very quickly. And one of the things that I encourage people to do is, as you think about all the different things that you could be doing, I mean, there's a million and one things, all of which are good things to be doing. You should go down a list of ideas that you have, of stuff that you could do, and say, all right, if this were to really work out, how important would it be? How many people can I affect? How many lives can we really help with this? And how much can we push the field forward? And use that as your selection criterion, in part, for what it is that you want to do.
There was some type of like symbiosis also that occurred in at least my situation, where I'm working with a basic science researcher that gave me an advantage to be able to work in this field of neuroscience. And I had the clinical aspect of it also that kind of drove a lot of what was happening in the lab that, he wasn't quite aware of, like things that I found seeing patients with smell loss and, like things, patterns, and so to speak, that we could look into further and build a mouse model on and things like that. So it was great working together in that respect, where we both had our own perspectives, not necessarily in a different scientific field, but just how we were going about looking at it.
Yeah. Well, I think, as we have discussed on other occasions, serving as a bit of a translator between the patient and the bench, clinician scientists can sometimes bridge that divide, and really, even with perhaps not a full-time commitment, but with a commitment towards understanding science that's happening in a basic lab, really positively contribute to bring relevance and patient impact to basic science. Most clinicians won't run their own lab, but they can certainly help to contribute to the overall scientific and translational mission of work by providing that input. Um, I want to just, being aware of the time, a couple of specific things that I did want to ask. And um, Rana, one of the things I would love to hear from you is from your perspective, what type of an individual should consider a career as an intramural clinician scientist? I think that that may be an area that some people are not necessarily familiar with, but there are some really exciting new programs I know from a training and career perspective that you may be able to touch on.
Thank you. So there, just like, to be honest, just like in regular universities, there's room for a lot of different types of clinician scientists also at the NIH. We do have a training program, and that is geared more towards people that want to develop as clinician scientists, have a track record in working in basic or translational science, but they just don't feel that they might be K Award ready immediately after residency. And they're looking for that additional mentorship, with the option of doing up to 2 days a week outside the NIH, of pure clinical work. And that is a program that Mike Ho on this call graduated, and Wei Chen and some other clinician scientists in other fields in otolaryngology. And it's a very successful program. We have a LASKER program, which is we're very much looking to recruit a first person for the NIDCD. And that's really for people that want to have patient-centered research and have a career around patient-centered clinical research. And that program offers support for a fairly long period of time at the NIH, and with the option of staying at the NIH later or transitioning to an extramural position. Other than that, the NIH has staff clinician positions. So that's for people that want to do more clinical work, albeit all the patients at the NIH are on clinical protocols. So for a patient to be at the NIH, they have to be on a clinical protocol. And we have our regular tenure track program where you have to be a competitive clinician, scientist, or scientist. So there's a wide variety, but I think that preferentially we're now looking to recruit people that want to do basic science that combines that has a strong correlation at least with clinical care.
Thank you. And Aaron, you bring a unique perspective as someone who now has a foot in industry. Are there specific skills that one needs in order to make that leap or to participate on the industry side in these really, really critical aspects of clinical trials and getting the bench research into patients?
Well, I will say just having a very rigorous clinical training is something that is always valued out in the world of industry, in particular in drug development. So making sure that you've got your clinical foundation solid, I think, is important. I do have to say, for anybody that's done that city training on research, which seems like it's super not that fun, you begin to live it every day. Once you're in the world of clinical trials. So actually read that stuff. It's important and useful to understand what the regulatory environment looks like out there, because it does very much inform the realities of how things get out there. But I think the biggest thing, to be honest, is if you pursue with passion the thing that you're interested in, if that thing happens to be something that is best done at industry, like making drugs, it will find you to a certain extent.
Thank you. Yeah, I think that sometimes that leap can seem a little bit intimidating, like maybe you need to have a business background or something. But I think your point is very well made, is that there are plenty of those people that are probably in industry, but they have it's uncommon that they have someone with outstanding clinical training and with context for the basic work that they're doing to help them to move their trials forward. And Susan, I wonder if you could just tell us, you mentioned that you got some early exposure towards public policy. You were in the DC area, but now working in health service research and being a true advocate for public policy on getting, both telehealth and in-person medical care to people in underserved communities. Any specific training that was necessary or steps that you took along the way to be so successful in that endeavor?
Well, I studied policy as an undergraduate at Princeton. I was in it used to be called the Woodrow Wilson School. I can't say that you necessarily have to have a degree in that. I think it's really boots on the ground experience, and working on the Hill was very influential because I started to understand the inner workings of that process and how to engage with lawmakers. But you don't have to have worked on the Hill to be able to be involved in policy. I think the most important thing is building relationships, because at the end of the day, it's all about people. And so if you're trying to work on a project that you ultimately want to make sure it can be sustained long term, and you're trying to develop relationships with lawmakers to make that happen, start working with them before you've ever submitted a grant. Build those relationships, involve them in the process, keep them up to date, get ideas from them because they will have good ones. Be open to adding new things to your studies. We have incorporated new cost effectiveness analyses and other things that policymakers asked us for, and had to be nimble and figure out how to do that, particularly if we were funded by Macquarie or entity that wasn't so fond of cost effectiveness at the time. But just being involved, I think, from the get-go and really developing those relationships can make a world of difference.
Yeah. Thank you. And Eric, in our last couple of minutes, one of the things that we see routinely in academic departments is that people who are successful clinician scientists get promoted to administrative positions. And administrative positions require their own amount of time for managing people and building departments. You serve as the division director of the rhinology division. How do you balance that? Does that end up taking away from other parts of your week?
Yeah, certainly. I mean, it is a time commitment. I'm very blessed that the people in my division are easy to get along with, and we are on the same page with most things. But there's always issues, not even with just like staff, but like ancillary, people in the clinic. It absolutely does. I mean, that's what I was saying about like how to juggle time. You have to be better and better and better at it as you progress with time, because you take on things that maybe you don't think about how much that actually is going to affect you. So yeah. I think one thing that I've learned is that when you take on something else, you should probably let go. And it's a good way of like mentoring other people and passing things on to junior faculty member to encourage them to, grow and establish themselves and get, good experience and help out. Altogether, I mean, I think, you should just be aware that there's certain time that you need to devote for your research. There's certain time that you devote for administrative duties, clinic, family, and you kind of have to keep those silos adjusted from time to time.
Absolutely. There are only so many hours in the day, although I think this group is probably very effective at squeezing the most out of them. Um, I want to end with a request to all of our participants. If you have thoughts or ideas about other specific topics you would like to hear from the Clinician Scientist Committee via webinars, if you could add any comments to the chat, that would be fantastic. I will keep the Zoom open so you can add those in. And I'm going to open it up to any other thoughts or considerations to add from our esteemed panelists or from the other members of the Clinician Scientist Committee.
Just speaking on balancing time, so thank you for each of the panelists for your time, and I learned so much from all of you. I'm glad it was recorded since I can watch it over and over again.
I'm sure we all will. Well, please add any thoughts that you have about future topics to the chat. And I want to, again, extend my deep thanks to all of our panelists. Thank you for joining us late in your evening. This is definitely cutting into something else you'd otherwise be doing, but we appreciate your willingness to share your experiences. So thank you all very much. And we hope to see you soon in Orlando.
Yeah.
Thank you, Aaron.
for moderating.
Yeah. Thank you for your excellent moderation, Aaron.
Thanks.